masuk lagi kan ke dah masuk sikit?
Dah masuk lagi.
Dah masuk lagi.
Lagi madam.
Alright, nice. Okay, nice. So topic 2, cell structure and functions. Okay, in SLT, as I said, as I briefed on table 4 SLT during introduction week, you need to spend 32. You need to spend 32 hours for topic 2. So 32 hours including the lecture, tutorial, practical, and also your independent learning. Okay, so the subtopics for chapter 2, 2.1 prokaryotic and eukaryotic cell, 2.2 structure and functions, cell membrane and organelles, 2.3 cell are grouped into tissue, 2.4 cell transport. Semua ni dah belajar kan masa SPM kan? SES of course lah dah belajar. SPM dah belajar kan masa ni? Benda-benda ni. So macam okey kot. Oh, boleh ke? Saya akan pergi laju sikit lah. Boleh ke? Dah belajar ke?
Dah madam.
Dah. Okay, nice. Alright. So, prokaryotic and eukaryotic cells state the three principles of cell theories, explain the structure on prokaryotic and eukaryotic cell, illustrate and compare. So, compare mesti ada apa? Hafizah? Kalau compare kena ada apa, Hafizah?
Ada persamaan dan perbezaan.
Yes, ada persamaan dan juga ada differences. Illustrate and compare the structure of prokaryotic and eukaryotic cells. Okay, so these are the cell theories. The first one, theory first, all organisms are made up of one or more cells. Number 2, new cells are always produced from pre-existing cell. Number 3, the cell is structural and functional unit of all living things. Number 4, the cell contains hereditary information which is passed on from cell to cell during cell division. Okay, so this is prokaryotic cell. Okay, the structure, the common structure of prokaryotic cell. Example of prokaryotic cell is bacteria. And what is the example of bacteria? Shafira Akilah, what is the example of bacteria? Shafira?
Yeah.
What is the example? Kenapa? What is the example? Seronok pula dia.
Saya tak tahu. Kejap saya
Okay, tak apa.
Google.
Tak apa. Tak payah Google. Kita tanya
Okay.
SES. Kita tanya SES. Sakinah, what is the example of bacteria?
E. coli.
E. coli, perfect. So example of bacteria is E. coli. E. coli, Staphylococcus adalah dua bakteria yang awak akan observe nanti masa SEM 2. Okay, eksperimen 1, SEM 2 awak akan tengok E. coli dengan Staphylococcus aureus. And kalau awak selalu suka minum Yakult atau Vitagen ataupun apa, ada lagi satu tu, tak famous sangat tu, dia ada bakteria baik iaitu Lactobacillus. Okay, so this is common structure. So common structure, apa dia ada? Dia ada flagella. Apa function flagella? Syamimi. Anis Nurjiha Syamimi. What is the function of flagella?
For movement.
Yes, very good. Betul. For movement, flagella is for plural, flagellum is singular. And then we have nucleus. Nucleus is not nucleus. Nucleus adalah tempat. Nucleus is the region where DNA can be found. Okay, and also a prokaryotic cell also have ribosome. But the ribosome for prokaryotic cell is smaller than eukaryotic cell. Of course, it has plasma membrane, it has cell wall, capsule, and pili. Pili, the function of pili is act as attachment to host or the other bacteria. Maksud attachment tu, contoh kalau kita ada prokaryot A, bakteria A dengan bakteria B. Okay, bakteria A dengan bakteria B. So dia nak connect kan macam mana, dia nak jumpa macam mana, dia akan connected melalui pili. Okay, pili. Pili ni ada pili yang biasa, ada sex pili. Kalau sex pili untuk reproduction, untuk tukar DNA from prokaryotic, from bacteria A nak pass kepada bacteria B. Okay, dia nak pass DNA. Itu untuk reproduction. And for bacteria we have yang ni lah, plasma membrane, cell, capsule. Basically bakteria ni dia ada 3 layer. Layer yang paling luar adalah capsule. Capsule ni dia adalah untuk protect bakteria. supaya bila white blood cell datang untuk proses phagocytosis, susah nak musnahkan. Okay, so the outermost layer is capsule, and then we have cell wall. Okay, the cell wall of prokaryotic cell is made up from Sharash. Cell wall of prokaryotic cell berbuat pada apa?
Peptidoglycan.
Betul, bukan gly tapi glycan. Okay,
Gly-
betul-betul. Tapi betul lah, okay, peptidoglycan. Kalau cell wall untuk plant diperbuat daripada apa? Hana? Hana?
Ya, ada.
Cell wall untuk plant cell diperbuat pada apa, Hana?
Selus.
Cellulose, very good, perfect. Okay, then third layer, barulah kita jumpa plasma membrane. Okay, so selalu kalau bagi soalan bagi gambar macam ni, make sure you know how to identify. Arrow tu dia menunjuk kat mana? Cell wall ke, capsule ke, plasma membrane. Okay, so this is the structure of prokaryotic cell. Prokaryotic cell no true nucleus. Kena ada perkataan true ni. Kalau dalam exam awak tak ada tulis true, awak tulis no nucleus saja, salah. Okay, no true nucleus. Maksudnya dia tak ada betul-betul nucleus. Nucleus ni memang tak ada. Okay, no membrane bounded organelle. There is no organelles that have membrane. For example, it do not has mitochondria. It do not has lysosome. It do not has chloroplast. Semua tak ada. Semua tak ada. Organelle yang ada membrane ni tak ada. And of course, it's very small. It also has DNA, but the DNA is circular. DNA dia adalah circular. And yang paling penting, DNA is not associated with histone protein. And what is the histone protein? Histone proteins looks like this. Okay, this is histone. Okay, histone protein. So this is the DNA for eukaryotic cell, for plant cell and animal cell. But for prokaryotic cell, okay, do not has histone. Tak ada, so histone ni semua tak ada. So dia punya DNA tu memang akan lie macam tu je. Dia akan baring macam tu, duduk macam tu je. Sebab tu kita panggil sebagai naked DNA. Okay, we call that as naked DNA, not associated with histone protein, or we call that as naked DNA. But in exam, you cannot write naked DNA because that is too general. Awak budak sains, so awak kena tulis not associated with histone protein. Okay, and the DNA is located in region called nucleoid. Okay, nucleoid. Alright, plasma membrane. Okay, plasma membrane. The function is to control the exchange of waste materials and nutrients. And then we have capsule. Okay, capsule will surround the cell wall. So macam saya cakap tadi, the outermost layer is capsule. And the function of capsule to protect the bacteria, okay, from phagocytosis by the host white blood cell. And then the cell wall, okay, as Sharash said, it is made up from peptidoglycan. Okay, peptidoglycan or murin. Okay, function dia of course cell wall untuk maintain dia punya cell shapes. The ribosomes smaller than in eukaryotic cell. Kalau ribosome for prokaryotic cell, the ribosome is made up from 70S, 70S. Dia punya unit tu adalah 70S. Kalau eukaryotic cell, 80S. Okay, the plasmid. Prokaryotic cell also has plasmid, and the function of plasmid to carry accessory gene. Okay, so the example of accessory genes is the genes that is resistance. Okay, the genes that resistant to ampicillin. So kalau dalam prokaryot ni, okay, tak tunjuklah dalam gambar ni. Tapi kalau dalam bakteria atau prokaryot, dia akan ada satu structure dikenali sebagai plasmid. Okay, bulat kecil je plasmid. And it's double stranded DNA. Okay, plasmid tu function dia adalah untuk carry accessory gene. Gene yang accessory, contoh accessory tu adalah gene that resistant to ampicillin. Okay, nanti awak akan belajar details lah dalam topik 8, recombinant DNA technology. Okay, for next structure, fimbriae, it has fimbriae or pili. Okay, kita tak sebut pili, kita sebut pilai. Okay, pilai. So function of membrane and pili is attachment to host or other bacteria. So for example, if the bacteria is parasite, the bacteria need host to survive. So the pili yang macam rambut-rambut kecil keliling dia tadi, so that pili is for attachment to host cell or for other bacteria, and also attachment to cell surface. Okay, Fimbriae is shorter than pili, so dia nanti kalau soalan bagi gambar kan, awak tengok kalau ada yang pendek tu adalah Fimbriae, panjang sikit adalah pili, and also ada sex pili. Sex pili adalah untuk transmit DNA between bacteria. So kita nak transmit, kita nak hantar DNA daripada satu bakteria ke satu bakteria lain melalui sex pili. And then we have flagellum singular, flagella plural. So kalau soalan bagi gambar, make sure you label dengan betul. Okay, flagellum function dia untuk movement. Cakap pasal label, saya dah tengok awak punya go formative. Okay, go formative untuk drawing tu. Tolong check balik tak ramai yang dapat perfect mark. So banyak sangat yang simple mistake lah yang awak buat. Okay, so tolong check balik. And then we have mesosome. Mesosome is the infolding of the plasma membrane. So this plasma membrane, bila dia fold macam ni, okay, dia fold, we call that as mesosome. And that mesosome, the function similar to mitochondria in eukaryotic cell. Okay, the function is similar to mitochondria in eukaryotic cell. Okay, untuk dia hasilkan energy untuk ATP, in the form of ATP. Okay, alright. Itu adalah prokaryotic cell. Kalau eukaryotic cell ada true nucleus. Okay, and DNA is enclosed by nuclear envelope. Okay, the DNA is enclosed by nuclear envelope. So the DNA is located inside the nuclear envelope. Okay, and the DNA is linear. Okay, it's not circular, it is linear and associate with histone protein. So macam ni lah. Inilah kita punya DNA. Associate with histone protein. Okay, contains membrane enclosed organelles such as mitochondria, chloroplast in the plant cell, Golgi body, ER we have two, we have smooth ER and rough ER, and then the lysosome in animal cells. Okay, and the cell wall in plant cell, the cell wall is made up from cellulose. In fungi, it is made up from chitin. Okay, chitin. So that is the structure of eukaryotes. So it also may have flagella or cilia. Okay, may have flagella or cilia. Constructed from a system 9+2 microtubule. Apa itu 9+2? Kalau awak tengok, dia ada 9 sini. 1, 2, 3, 4, 5, 6, 7, 8, 9. Plus 2 tengah-tengah. Okay, 9+2 microtubule. Okay, dia punya arrangement tu lagi kompleks. Microtubule punya arrangement tu kompleks untuk make up the flagella ataupun cilia. Kalau untuk prokaryotic cell, dia ada simple flagella atau dia ada simple flagella sahaja. Okay, dia tak arrangement dia bukan macam ni. Okay, bukan 9+2 ni. Alright. Okay, so comparisons. Okay, comparison between prokaryotic and eukaryotic cells. So comparison between prokaryotic and eukaryotic cells. So similarities, both cells surrounded by plasma membrane. So dua-dua sel prokaryote and eukaryotic surrounded by plasma membrane. Okay, and both cell contain DNA. Tak kisahlah DNA linear ataupun circular, tapi dua-dua tu ada dia punya DNA. Alright, so setiap satu point dapatlah satu markah comparison. Okay, always include the similarities. Okay, kalau awak ingat differences, awak ingat differences, 8 differences awak ingat. Tapi similarities awak tak letak, still awak tak akan dapat full mark. Okay, so the differences, prokaryotic cell, no true nucleus. Eukaryotic cell, true nucleus is present. Okay, DNA is not bounded by nuclear envelope. Eukaryotic cell, DNA is bounded by nuclear envelope. Okay, ingat true tu kena underline. Wajib tulis. So better, I think it's better for you to underline or highlight the word, the true, okay, yang perkataan true tu, underline or highlight dalam awak punya lecture note. Okay, prokaryotic cell, the difference number 2, DNA molecule is circular. Okay, while eukaryotic cell is linear, not associated with histone protein. Associate with histone protein. Okay. Yang ketiga, no membrane bounded organelle. Membrane bounded organelle are present. Ada. Benda yang dalam bracket tak payah cerita. Tak payah cakap bagi example. Cukup sampai sini. Full stop. Okay. Number 4, smaller. Larger. Bila awak cakap smaller, awak kena state dia punya berapa size tu. Same goes for eukaryotic cell. Kalau larger, kena cakap bigger than 10, berapa? Bigger than 10 micrometer. Okay, untuk point yang ni, saya nak bagi tahu sikit. Untuk point yang ni, ada satu tahun tu, I can't remember when, I think it was 3 to 4 years ago. Okay, soalan bila dia suruh, tahu bila tak ingat, lupa. Soalan bila dia bagi macam ni, dia tanya differences prokaryotic and eukaryotic cell. Kalau awak cakap smaller, larger, salah. Ada satu tahun tu, dia kena bagi spesifik berapa saiz dia. Dia tak terima less than 10 micrometer, larger than, bigger than 10 micrometer. Tahun tu lah, masa tahun tu dia tak terima, dia kena bagi exact size berapa. So my suggestion untuk point number 4 ni, awak letak yang paling bawah lah. Okay, paling bawah. Yang tak berapa nak penting sangat. Okay, saya punya suggestion lah. Okay, nak selamat, awak ambil point-point yang penting. Right? Okay, question. Differences number 5, ribosome are smaller. So as I said, 70S. For eukaryotic cell, larger, 80S. Okay, and then you can also discuss about cell wall. Cell wall prokaryotic cell is made up from peptidoglycan, while for eukaryotic cell, it is made up from cellulose in plant cell and chitin in fungi. Okay, and the next one, some have simple flagella. Simple, dia tak complex. While eukaryotic cell have 9+2 microtubule arrangement. And the last one, for prokaryotic cell divide through binary fission. For eukaryotic cell, divide by mitosis or meiosis. Okay. What is the example of eukaryotic cell? Anas.
Yeah.
Anas, example of eukaryotic cell.
Eukaryotic.
Yeah.
Animal cell.
Betul? Animal cell lagi satu? Lagi satu?
Plant cell.
Yeah, plant cell. Animal cell, plant cell. Betul, pandai. Okay, and then we move on to the next one. Structure and functions, cell membrane and organelle. Okay, kita akan cover A, B, C, D. Okay, A, B, C, D. Show the detailed structures of typical plant and animal cell. Allah, ini dia belajar daripada sekolah menengah. And state the organelle present. For B, explain the structure and function of the following organelles: nucleus, rough ER, smooth ER, Golgi body, lysosome, ribosome, mitochondria, chloroplast, and centriole. So all the organelles in learning objective B, you have to remember the structure and also the functions. Okay, structure and function kena ingat. Okay, bukan ingat hafal je structure tapi awak kena tahu gambar. Rupa dia macam mana. Tipulah kalau tak tahu mitokondria macam mana rupanya. Ni dah belajar dah. Alright, animal cell and plant cell. Okay, animal cell it is bounded by plasma membrane. Okay, plasma membrane, the membrane envelopes a jelly-like cytoplasm which contain nucleus, organelles such as mitochondria, endoplasmic reticulum, Golgi body, centriole, lysosome, and ribosome, and the other cell components such as cytoskeleton, a pair of centriole. So animal cell ni ada plasma membrane, tak ada cell wall. Okay, and also have does not have chloroplast and plastic. Okay, plastik ni macam chloroplast. Okay, tapi dia tak ada main, dia bukanlah main organelle. Okay, dia bukan main organelle. Dia macam chloroplast tapi dia bukan main organelle. Okay, animal cell dia ada vacuole, tapi vacuole dia no lush. Vacuole dia okay, it's not large or permanent central vacuole and tonoplast. Only temporary tiny vesicles or vacuole. So dia tak adalah vacuole yang permanent. Macam plant cell vacuole dia permanent, besar, and then ada tonoplast. For animal cell, only temporary tiny vesicles or vacuole. Animal cell awal je ada tiny vesicle ataupun vacuole. As it grows up, as it become larger, dah tak ada benda tu. Okay. And this is animal cell. This is diagram, the diagram from Campbell. Okay, buku Campbell. Yang ni lah gambar. Okay, ini adalah animal cell. Alright, boleh tengok lah. Plant cell. Plant cell, the unit structure possessed only by the plant cell that differ from the animal cell. Plant cell ni ada a few things yang sama macam animal cell, tapi yang bezanya dia ada cell wall. Cell wall is made up from cellulose that gives shape and mechanical support or strength to the cell. And also have plasmodesmata. Plasmodesmata adalah tiny opening at the cell wall. Okay, it's like pintu, it's like a door. Okay, dia macam pintu dekat cell wall tu. Okay, pintu tu untuk untuk connectkan satu sel kepada satu sel. Siapa yang nak connectkan satu sel kepada sel ada pintu. Pintu tu, okay, laluan pintu tu adalah plasmodesmata. Okay, apa function dia? It allows the movement of ions and small molecules between cell. So ion and small molecule bolehlah bergerak macam tu. Air pun boleh bergerak lalu kat situ. Okay, tak ada gambar plasmodesmata. This is plasmodesmata. Okay, yang tu. Okay, dia macam small channel atau small opening at the cell wall. Allow the molecules, ions and also water to pass through. Okay, that is plasmodesmata. And what else do we have here? Okay, chloroplast. Of course, plant cell ada chloroplast. Kalau tak, macam mana dia nak buat glucose? Okay, chloroplast also has other type of plastic, leucoplast, chromoplast. It has large permanent central vacuole bounded by tonoplast. Okay, dia ada vacuole yang besar ni. Yang ni. Large permanent central vacuole and bounded by tonoplast. Maksudnya dia punya membrane tu kita panggil dia sebagai tonoplast, bounded by tonoplast. Okay, and the store food is mainly starch. Okay, dia akan store food in the form of starch, lack of centriole, lack of lysosome. Okay, so this is plant cell. As you can see, it has nucleus, same as animal cell, rough ER, smooth ER, Golgi apparatus. For Golgi apparatus, kalau nak tulis perkataan Golgi tu, G tu wajib huruf besar. G huruf kecil salah. Last 2 years before COVID. COVID bila 2019. Okay. 2018 kot. Entah. 2018 ke? 2019. 2019. Sem 1. Bila marking ada student tak tulis huruf G ni huruf besar. So dia tulis macam ni, Golgi body. Salah. Macam ni kena betul. Okay. And then we have mitochondria. The plasma membrane, the chloroplast, okay, the cytoskeleton, which is microfilament microtubules. Okay. So this is the differences between animal cell and plant cell. Soalan bukan saya tak nak, bukan macam nak cakap soalan senang macam ni takkan keluar, tapi macam dari dalam chapter 2 ni banyak benda yang boleh ditanya. So soalan senang macam ni macam rarely keluar lah. Okay, rarely keluar dalam exam. Okay, sebab dia sangat-sangat senang. Awak dah belajar daripada sekolah lagi dah. Okay, so you can see these are the differences of animal cell and plant cell. We have how many? 3, 5, 9. Yeah, we have 11. Okay, we have 11 differences. Each difference count as 1 mark. Okay, setiap satu difference adalah satu markah. Okay. Alright. Organelles. Okay, and then we have organelles. Organelle, apa yang awak kena tahu untuk organelle ni? Awak kena tahu structure dan juga functions. Okay, structure and functions. Okay, structure and function. So, are we going to discuss, maybe we're going to discuss until, kejap mungkin kita akan discuss sampai smooth ER je kot. Okay, nucleus, rough ER and smooth ER. Okay. Okay, nucleus. Nucleus adalah double membrane bounded organelle. Okay, double membrane bounded organelle. Maksudnya dia adalah organelle yang ada double membrane. Outer, inner. Outer, inner. Ada tiga organelle yang ada double membrane. Apakah organelle tersebut? Alia Syakira. Bagi saya satu. Nucleus dah. Bagi saya satu lagi. Organelle yang ada double membrane ada 3. Nucleus dah. Lagi 2. Bagi saya satu. Alia Syakira.
Plasma membrane.
Kan? Plasma membrane mana ada double membrane? Jaslina.
Mitochondria.
Sorry?
Mitochondria.
Very good. Mitochondria, nucleus. Lagi satu. Sarah Sofia.
Vocal.
Macam saya cakap tadi, vacuole dia ada tonoplast. Vacuole ada tonoplast. Dia tak ada double membrane. Keying. Satu lagi.
Chloroplast.
Yes, very good. Chloroplast. 3 organelles that have double membrane. Nucleus, mitochondria, chloroplast. 3. Okay. Alright. So, first organelle structure, the nucleus, double membrane bounded organelle. Average size is 5 micrometer in diameter. Contain most of the genes. Okay, so gene memang duduk dekat situ lah. Yang tu lah kita akan pas tu. Alright, and it consists of nucleolus, nuclear envelope, nucleoplasm, and chromosome. Okay, bila dia dah condense and thicken, we call that as chromosome. Bila dia lengthen and decondense, kita panggil dia sebagai chromatin. Okay, so between nucleolus, nuclear envelope, nucleoplasm, which structure that we can found the DNA, Anas?
Ya.
Antara nucleolus, nuclear envelope dengan nucleoplasm, kat mana kita boleh jumpa DNA?
Nucleolus.
Very good, nucleolus. Okay, nucleolus. Okay, so nucleolus, this is nucleolus. Okay, this is nucleolus. Okay, yang region, tak nampak Okay, nampak. Tu nampak. Okay, nucleus yang tengah-tengah ni. Okay, dia adalah spherical structure in the nucleus. Bayangkan nucleus tu kecil, dalam dia ada lagi yang kecil. iaitu nucleolus. Yang dalam dia ada DNA. Dalam lagi DNA daripada ada nucleotide. Okay, so lagi dia sangat-sangat kecil lah. So bayangkan DNA yang sangat-sangat kecil ni boleh bawa banyak information, okay, untuk pas kepada sel-sel seterusnya. Okay, so it appear as mass of densely stained granules and fiber. It contains DNA, RNA, and proteins. So dia ada DNA, RNA, protein. So di DNA ada apa? Ada genetic information untuk synthesize rRNA. Okay, so as you can see, this is nucleus. Dia ada juga nuclear pore. Nuclear pore, lubang dekat nuklear tu. Okay, lubang dekat nukleus tu. So bila dia dah hasilkan protein semua kan, dia akan keluar melalui nuclear pore. Okay, dia akan exit melalui nuclear pore. And then we also have nuclear envelope. Okay, macam envelope, sarung lah, sarung nukleus tu. Okay, and then the endoplasmic reticulum. Alright, the nuclear envelope encloses the nucleus, consists of two membranes. Okay, consists of two membranes, the outer and the inner membrane. Okay, outer dan inner membrane. Alright, and nuclear envelope is perforated by pore. So maksudnya dia ada dekat surface nuclear envelope tu, dia ada nuclear pore. Okay, function to regulate the movement of molecules into and out of the nucleus. So dia akan mengawal lah siapa yang masuk dan siapa yang keluar. Okay. And nucleus is connected by ER, okay, endoplasmic reticulum. Specifically, dia akan connected by rough ER. Okay, rough ER akan duduk dekat lagi dengan nucleus. And the function of nucleus contain genetic material of the cell. Okay, dia ada genetic material of the cell. Dia ada center, okay, act as the center to control cell activities and cell division, production of RNA, mRNA, tRNA, rRNA. Okay, so nucleus ni memang dia macam, nucleus ni basically is like brain of the cell. Okay, kalau macam kita punya brain ni adalah benda yang paling penting. Kalau brain kita brain dead kalau orang panggil kan, kalau dah brain dead tu memang mati lah, tak boleh buat apa lah. Sama macam nucleus. Nucleus, kalau nucleus tu dah tak ada function, habislah sel tu. Habislah sel tu. Sebab tu kalau untuk immunity, sesetengah immune system, dia akan attack nucleus dulu. Okay, dia akan attack nucleus dulu. Siapa ada tengok anime Cells at Work? Ada tak tengok anime Cells at Work? Sakinah tengok, Sakinah?
Ada. Tengok.
Tengok kan? So tengoklah kartun anime yang memberi manfaat. Okay, seperti Cells at Work. Okay, awak tengok kan, kalau macam white blood cell tu kalau dia rasa macam ada je foreign cell, dia punya topi tu akan naik. Okay, macam ada topi, macam isyarat lah, macam signal, topi dia akan naik. Macam ada benda masuk. Ada benda foreign lah yang masuk ke dalam sel. Okay, so menarik lah anime tu untuk tengok. So siapa yang tak tengok boleh lah nak tengok. Kalau siapa yang tak minat anime tak apa, okay je, tak paksa. Tapi that is just example. Okay, the endomembrane system. Soalan ni pernah keluar quiz. Lah. Quiz je kot. Yes, quiz. Kita buat sebagai macam midterm punya test lah. Midterm punya test. Soalan ni pernah keluar. Endomembrane system. And malangnya ramai tak dapat jawab. Bukan batch awak, batch lepas. Okay. The endomembrane system, apa itu endomembrane system? Many of different membrane of eukaryotic cell. Okay. Related through direct physical continuity or by the transfer of membrane segments such as tiny vesicle. Maksudnya, dia akan ada different organelles yang ada membrane ni akan bekerjasama untuk direct physical continuity ataupun untuk transfer of membrane ataupun untuk transfer, untuk membawa satu vesicle daripada satu tempat kepada satu tempat yang lain. Okay, so endomembrane system tu siapa yang terlibat? Okay, yang ni lah budak hilang markah batch lepas. Dia tak boleh bagi siapa organel apa yang terlibat dalam endomembrane system. So endomembrane system yang terlibat, organel yang terlibat adalah nuclear envelope, rough and smooth ER, transport vesicle, Golgi body, lysosome, secretory vesicles, and plasma membrane. Okay, itu adalah yang terlibat. Organel yang terlibat adalah benda ni. Okay, sebab plasma membrane is not organelle. Okay, plasma membrane is not organelle. So rough ER, okay, rough ER, where is it? Rough ER, yeah, this one is rough ER. And we also have smooth ER, where is it? Yeah, this one. And then the transport vesicle, this thing, the Golgi body or Golgi apparatus, the lysosome, yeah, this is lysosome. And then the secretory vesicle. So these organelles work together, okay, and to transfer the membrane, okay, to transfer of membrane segments, okay, such as tiny vesicle. So diorang ni bekerjasama lah, start daripada nucleus, rough ER, smooth ER, transport vesicle, Golgi body, lysosome, and then secretory vesicle untuk bawa keluar. Untuk bawa keluar. Kalau dia nak bawa keluar, nak pas pada sel lain, dia akan keluarkan macam ni lah. Dia akan secretkan kepada luar by the process of exocytosis. Okay. Relationship among organelles of the endomembrane system. So ini adalah relationship dia lah. Okay. Awak boleh baca kot. Boleh baca. Benda yang sama. Endoplasmic reticulum. Okay. Ataupun ER. Rough ER, smooth ER. Saya tak silap saya, kalau dalam exam boleh tulis ER. Okay, ER boleh tulis. Setakat yang kita discuss yang terbaru, ER boleh, endoplasmic reticulum boleh ditulis sebagai ER. Okay, tapi awak tak boleh tulis RER. Salah. Kena tulis penuh. Kalau nak describe rough ER, kena tulis rough, bukan awak tulis R saja. Okay. So endoplasmic reticulum, the largest of the internal membrane. Okay, most extensive portion, continuous with outer membrane of the nucleus. ER includes membranous tubules or sac, ER lumen, the internal spaces. So we have two types of ER, rough ER and smooth ER. So this is a rough ER. Okay, smooth ER. So between rough ER and smooth ER, what are the things that you can differentiate, ataupun apa yang boleh nampak daripada gambar ni, apa yang boleh nampak perbezaan antara rough ER and smooth ER? Jude Rush.
Yes, madam.
Yeah, what's the difference between rough ER and smooth ER from this diagram?
Rough ER has the ribosomes on it, but smooth ER doesn't have ribosomes.
Okay, very good. Ribosome, ada ribosome untuk rough ER, tak ada ribosome untuk smooth ER. And then Shafri, lagi difference. Apa beza rough ER and smooth ER?
Rough ER ni dia macam panjang-panjang sikit sebagai tarik.
Panjang. Okay. Rough ER panjang-panjang. Okay, smooth ER pendek-pendek.
Bulat-bulat.
Bulat-bulat. Okay, boleh bulat-bulat. Okay, good. Boleh try. Good try. Lagi siapa lagi? Carmila.
Yes, madam.
Iya, Carmila. Apa lagi difference?
Function of both ER.
Okay, you cakap function. Give me one function of rough ER.
Rough ER dia to pack and transport protein.
Okay. Smooth
Untuk
ER.
smooth ER pula function dia untuk dia synthesize lipid.
Okay, good. Very good. So, 3 lah. Okay, ada banyak lagi yang actually you can discuss. Okay, macam siapa cakap tadi? Anas Shafri cakap, Shafri ke yang cakap panjang-panjang pendek-pendek? Okay, rough ER flatten. Dia ada flatten shape. Okay, flatten. Kalau untuk smooth ER tubular. Okay, tubular macam bulat-bulat macam Syafri cakap. Okay, flatten, tubular shape. Rough ada ribosom. Jude Rush cakap ada ribosom. Smooth lack ribosom. Okay, and rough continuous from outer membrane of nuclear envelope. So basically, rough dia lebih dekat dengan nukleus. Smooth lebih jauh daripada nukleus. 3 benda tu yang you boleh differentiate ataupun you boleh tengok. Okay, atau ya, daripada kalau soalan bagi gambar kan, okay, you nak identify yang mana? Tengok 3 benda tu. Shape, ada ribosom atau tak, lepas tu location. Right? And rough found in many in cells that synthesize protein for export. For example, hormone digestive system, digestive enzymes and antibodies. So this is the function of rough Rough is to pack and transport protein synthesized by ribosomes attached to the Jangan jadi malas. Okay, macam dalam go formative tu malas. Tak label pun. Nak tunjuk removal of water pun malas. Okay, tulis penuh. To pack and transport protein synthesized by ribosome attached to the Okay, smooth apa dia punya function? Synthesize lipid, involved in carbohydrate metabolism, detoxifies drugs and poison, store or release calcium for muscle contraction. Okay, rough smooth Okay, ada soalan setakat ni? Ada soalan ke? Ada soalan tak nak tanya?
I have a question.
Yes, Kritina, what is it?
The ribosomes part at the very first starting, the prokaryotic cells. Madam, you said something about 70S. What does that mean? What does that S mean?
It's the unit. Actually, for example, kalau macam kita air 100, 1000 ml, 1 liter. So S tu adalah dia punya unit. Ribosom punya unit. S.
Okay. And then another one I don't understand is the 9+2 microtubules.
Alright, good question. Okay, 9 plus 2. This one, right, Kitina?
Yeah. That's okay.
Flagella have 9 plus 2 microtubule. Okay, as you can see, mana dia? Yeah, I just erase all the ink. Okay, so a 9 plus 2 microtubule refer to the arrangement of microtubule. So you can see, okay, kalau tak clear gambar ni, kita tengok gambar ni. Okay, 9 plus 2. Dekat mana 9 plus 2 tu? Setiap ni, okay, 1, 2, 3, 4, 5, 6, 7, 8, 9 plus 2. So dia arrange dalam bentuk bulat and ada tengah-tengah ni lagi 2. Itu adalah 9 plus 2 microtubule. Microtubule tu adalah protein. Protein yang made up the flagellum. Okay, so how it is arranged? It arranged like this. In pair, tapi keliling dia ni ada 9. Okay, keliling tu dia ada 9. Keliling dia ni ada 9. Okay, semua duduk berpasangan. Pair. And at the center ada lagi 2. Yang tu. Boleh ke? Dia 6 plus 2 microtubule ni refer pada arrangement, okay, Tina. Okay, alright.
Okay.
That is the arrangement for the complex flagella untuk eukaryotic cell. Kalau untuk prokaryotic cell, dia simple saja, dia tak arrange macam ni. Okay, lagi ada tak soalan? Ada lagi tak?
Madam,
Yeah.
Can you explain the endomembrane system? Like if they ask you what is the endomembrane system, then how do we put it in words?
Okay. What is endomembrane system? Let me check first. Alright, what is the membrane system? Okay, what is the endomembrane system? Okay. Okay, kalau you nak cakap what is the endomembrane system, you explain this point. Okay, yang ni. Point this. Sekejap saya padam dulu. Okay, clear. Alright, you explain this point. You define what is endomembrane system, and then you state what are the organelles involved. Okay, organelles involved. And then itu sajalah. Define and then state the organelle involved. Yang tu.
Okay, thank you, madam.
Welcome. Last time dia keluar sebagai soalan structure, dia tak keluar sebagai essay pun. Okay, dia keluar sebagai structure. Bukan dalam PSPM, sorry, not PSPM, dalam quiz. Macam midterm test saya cakap tu, dia keluar sebagai soalan structure. Okay, ada lagi tak soalan lain? Lagi tak? Tak ada. Sure tak ada? Okay, so we're going to continue on Thursday. Okay, on Thursday. On Wednesday, we're going to do experiment 1, basic technique microscopy. Alright, and then okay, untuk Amira Alia. Tak Amira Alia.
Ya, saya.
Okay, Amira Alia, kenapa awak ke yang record tadi?
Tak.
Kenapa dia masuk dalam awak punya?
Tak
Awak record ke tadi?
pun. Tak record
Okay.
madam.
Nanti bagi saya